Good morning, students. So, this is the part 3 of our chapter Cell, the Unit of Life. So, detailed structure of cell membrane was studied only after the advent of electron microscope in 1950. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells, enabled the scientists to deduce the possible structure of plasma membrane. So these studies showed that cell membrane is composed of lipids that are arranged in a bilayer form. Also, the lipids are arranged within the membrane with the polar heads towards the outer sides and hydrophobic tails towards the inner side. Hydrophobic and hydrophilic, I think all you know. Hydrophobic means water repelling and hydrophilic means water loving. So this ensures that the non-polar tail of saturated hydrocarbon is protected from aqueous environment. The lipid content of membrane mainly consists of phosphoglycerates, and in this bilayer of lipid there is present protein. Intrinsic and extrinsic proteins are also present there. So later Singer and Nicholson did the made a model of fluid mosaic model, and we'll study it in next slide. This slide shows the model of cell membrane that is called fluid mosaic model by Singer and Nicholson. Previous to this model, many models were given by scientists. It was given by Gotter and Grindel model, then Daniel and Devson model, and then Robertson model. But all these models have some drawbacks. Later on, it was accepted. Fluid mosaic model by Singer and Nicholson was accepted by all. So in early 1972, Singer and Nicholson proposed fluid mosaic model for the membrane structure. According to this model, the biomembrane is not solid but viscous fluid, and the phospholipid and protein molecules are arranged not uniformly but as a mosaic. As per the fluid molecules having globular proteins associated with them. So the lipids and proteins vary from one type of membrane to another. The cell membrane also has oligosaccharides on the exposed surface. A good amount of cholesterol is also present in the cell membrane of the animal cells. So phospholipid molecules are amphipathic, means they have both hydrophilic and hydrophobic regions. They have polar hydrophilic heads directed outward and non-polar hydrophobic tails pointing inwards. This arrangement forms a water-resistant barrier which allows only liquid materials to pass through it. The phosphorylated bilayer is 35 Armstrong thick. The two layers have different kinds of lipids. The lipid molecules can move sideways in this layer, changing places with their neighbors. This property is called fluidity, allows the membrane to stretch under stress and to reseal itself if it is disrupted. The membrane's fluidity also allows the proteins to move within it. Inside the lipid bilayer, you can see proteins are embedded. These proteins are called intrinsic proteins. With the proteins which are present inside the lipid bilayer, between the lipid bilayer, they are called intrinsic proteins, and which are present outside, they are called extrinsic proteins. So the extrinsic proteins are peripheral proteins, and the proteins which are present inside the bilayer membrane, they are called integral proteins, or tunnel proteins are also called they, and they are having oligosaccharides. The oligosaccharide molecules usually branched and are present on the exposed surface of cell membrane. They are associated with protein as well as lipid molecules, respectively forming a glycoprotein and glycolipids. So, some molecules of cholesterol are also present. Cholesterol molecules are inserted between the phosphorylated molecules of plasma membrane of animal cells. They are more stiff than phospholipids and therefore help stabilize the membrane. Some bacteria contain pentacyclic sterol, and they are called just like hapanoids. In the cell membrane, the hypenoids play the same role as the cholesterol. So this is all about, this is very important diagram, and you have to draw this very carefully, and this is called Singer and Nicholson fluid mosaic model of model of plasma membrane. As I have earlier told you, that fluid mosaic model means it provides fluidity. That integral proteins which are present, they help the substances to move from one place to another place. So according to this, the quasi-fluid nature of lipids enables lateral movements of protein within the overall bilayer. This ability to move within the membrane is measured as fluidity. Now you must know that the in human beings the membrane of erythrocyte has approximately 72% protein, 52% protein and 40% lipids, and rest of are 8% are oligosaccharides. So here are the functions of plasma membrane. One of the most important function of plasma membrane is the transport of molecules across it. The membrane is selectively permeable to all molecules present on either side of it. Selectively permeable. We have studied about selectively permeable in ninth standard also. So many molecules can move briefly across the membrane without any requirement of energy. And this type of movement is transport is called passive transport. When no expenditure of energy is there, that will become passive transport. Neutral solutes may move across the gradient, that is from higher concentration to lower. Water may also move across this membrane from higher concentration to lower concentration, and movement of water by diffusion is called osmosis. Osmosis, you all know. We'll do the diagram when the class will start. Practicals will start on the osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require certain protein of the membrane to facilitate their transport across the membrane. If few ions or molecules are transported across the membrane against their concentration gradient, means against the concentration gradient means from lower concentration to higher concentration. Such a transport is an energy dependent process in which ATP is utilized, and it's called active transport. Means active transport means with the expenditure of energy and from lower concentration to higher concentration. Just like sodium and potassium pump.